Well, welcome everybody to this new DaSCI seminar. Today it's my great pleasure to introduce Professor Karsten Borgwardt, Director of the Department of Machine Learning and Systems Biology at the Max Planck Institute of Biochemistry in Martinsried, Germany. His work won several awards, including the €1 million Krupp Award for Young Professors in 2013, and a starting grant 2014 from the ERC backup scheme of the Swiss National Science Foundation. Professor Borgwardt has been leading large national and international research consortia, including the personalized Swiss sepsis study and the subsequent national data stream on infection-related outcomes in Swiss ICUs, as well as two Marie Curie innovative training networks on machine learning in medicine. In this talk, he will describe their efforts towards data-driven early recognition of sepsis and the related problem of antibiotic resistance prediction. I want to thank, first of all, Professor Borgwardt for very kindly accepting our invitation. And whenever you are ready, the floor is all yours.
Thank you very much, Pablo. Thank you for introducing me. Thank you for this kind invitation to present my work here. So I will tell you now in the next hour about our work on machine learning in medicine, and I will do so by highlighting two efforts of ours, namely predicting sepsis, which will take the major part of this hour, and then also summarizing our efforts in antibiotic resistance prediction using machine learning. Now, sepsis prediction is a very fundamental topic in machine learning in medicine. I want to convince you of this statement now in the following. But in order to convince you, let's first define the need for predicting sepsis. Now, what is sepsis, first of all? It's a global health crisis. There are around 50 million cases per year, and there's one death from sepsis every 2.8 seconds. And even if you survive, you may face lifelong consequences. And 1 in 5 deaths worldwide is associated with sepsis. And on top of that, it's the major cause of death in hospitals worldwide, and in particular in intensive care. Now, what is now this phenomenon, this syndrome of sepsis? So sepsis arises, to define it a bit informally, when the body's response to an infection hurts its own tissues and organs to a degree that it results in what is called a septic shock, then single organ failure, multi-organ failure, and ultimately, in many cases, even death. So these elements are very important. So it's like an immune response to an infection. These are the two criteria, and the third one is that injures the organs and the tissues of the patient. These three things come together and then cause extreme harm to the patient. There are symptoms of sepsis like slurred speech, extreme shivering, passing no urine, or other like weak indicators of sepsis that are shown here on this slide. But as I just said, they are only weak indicators in a machine learning sense. So none of these is a highly precise predictor for sepsis. So you have weak indications of sepsis, but not strong like predictors. So after this more informal introduction of sepsis, let's now turn to our attention to the formal definition, the formal international medical definition of sepsis that is called sepsis 3. It's the international consensus definition, and it includes the same elements I described before, namely that sepsis is a life-threatening organ dysfunction, so it harms organs and tissues, caused by a dysregulated host response, that's the second element, to an infection. That's the third element. And why is it now relevant to detect sepsis early? Well, sepsis is not a condition that is without any treatment, or that we cannot in many cases cure. In fact, you can often treat this infection that is at the heart of all of this using antibiotics. But in order to do an effective antibiotic treatment, you have to give an antibiotic that is tailored to the infection that you have, to the particular species that is behind the infection in a given patient. Identifying the bacterial species that is responsible for infections in blood still takes between 24 and 48 hours. However, this time delay in identifying the bacterial species increases mortality. Each hour of delayed effective antibiotic treatment increases the mortality in intensive care. So therefore, from a medical perspective, it's extremely important to detect sepsis early, to be able to start the antibiotic treatment against the pathogen that is potentially causing the sepsis early. So we have here like a prediction problem of the highest clinical importance and of the highest clinical interest. Now, this is actually nicely summarized in a quote by Niccolo Machiavelli from Il Principe. He refers to a similar condition, not exactly sepsis, but to hectic fever, so a related condition. And he says about hectic fever at its inception is difficult to recognize, but easy to treat. Left unattended, it becomes easy to recognize and difficult to treat. Now, the role of machine learning, of data science, now in this context is to detect sepsis, not hectic fever, but sepsis early at a time point when it's still rather easy to treat, but so far for the human doctor, difficult to recognize. That's the role of machine learning in this context. And now to describe in an abstract manner what we do, what we and others do with machine learning algorithms now in this context is that we take data from the intensive care unit. That's like a common place where sepsis patients end up. We take the data-rich environment from intensive care that records vital signs of a patient, health record, health history, treatment in the ICU, lab test results, and so on. Here in this example view, I only mention a few vital signs. So we take these vital signs, so time series that are recorded over time that describe some vital properties of the patient. And then looking at these time series up until point X, we predict whether that patient will develop sepsis. Will the patient develop sepsis? When will the patient develop sepsis? Will the patient get a severe form of sepsis, or septic shock? Will the patient even die from sepsis in the future, within a certain time window in the future? Like, one thing we have to think about or define is how big is that time window for which we make the prediction. So that's our typical setup. Intensive care data go into our algorithm. We predict a sepsis-related phenotype as our output. As I mentioned, the ICU is particularly interesting for that, because there we really record a lot of data about a given patient. So the patient is intensively, as the name already suggests, observed and supervised by the doctors and the machines in these ICUs. Now, there are two main machine learning approaches or concepts that my lab has been working on in this context. I want to mention the first one, and I want to discuss in much more detail than the second one. The first one is maybe more of a specialized approach that is not so common in the field, but that we took great interest in. Namely, we tried to discover what you could call digital biomarkers in these patient time series. So is a particular trajectory in one of these time series of the vital signs, is a particular shape of one of these time series associated with later on developing sepsis. If you know a bit about genetics, you will understand the analogy to genome-wide association studies, where you try to find single nucleotide polymorphisms, so genetic variants in the genome that are whose presence, occurrence, is associated with a phenotype of interest. And here we do something similar, just not with genetic data, but with these time series data that we have, trying to find sub-time series, so intervals within these time series, that have a particular shape and that are associated with developing a particular phenotype, in our case, with developing sepsis. And there we have published papers on this type of association mapping in time series. We call it shapelet mining in bioinformatics. Bock et al. And Gumsch et al. Are here the two key references in which we presented this work. The second approach is the much more common one. That's the one I described before. That's a classification approach where you look at all the data that you have recorded about a patient up until time point X, and then you try to predict whether that patient, now given all the information that is available at time point X, will develop sepsis in the future. That's what we call the classification approach to sepsis prediction. So it's a binary classification. Will this patient, in simplest scenario, this is a binary classification. Will this patient develop sepsis? Yes or no? This has been another like key topic in my lab. Now let's have a closer look at how this prediction of sepsis through time series classification can be done. In fact, when we turned to this problem in 2017, 2018, we were by no means the first ones to do that, to predict sepsis using classification approaches. We discovered, as you will see in a minute, more than 20 papers that had already tried to do that. But it was very unclear how these different approaches that had been proposed there, let's say over the last 10 years, how they compare to each other. Which one is the best out of them, and best in which sense? How can you compare them at all? Do they have common challenges and pitfalls? These were all like questions that we asked ourselves when we embarked on this new line of research in my lab, something like 5 to 6 years ago. And in order to answer this question, we performed a systematic review of this literature, and this was spearheaded by Michael Mohr and Bastian Rieck, who shared the first authorship on this paper, Frontiers in Medicine 2021. So what did we do in this systematic review? So we looked at all studies that had used machine learning for sepsis prediction before. We ended up with 22 papers, 21 of which really presented a new method for this task, or a new approach for this task of sepsis prediction. And the 22nd one presented a new data set for this task. And all the ones that present a new approach are retrospective studies that work on data sets that have been collected before. 12 out of them use the MIMIC database. That's an intensive care database from Massachusetts. So more than half of them. 12 out of these studies use sepsis 2. That's the predecessor definition of sepsis 3. 9 use sepsis 3 as we defined before, and 1 study from Heidelberg used sepsis labels annotated by clinicians. So you can already see there's some diversity even in how they define sepsis. So 3 different ways of defining sepsis. So there we have a lot of diversity. We have very much homogeneity in terms of the data set that's being used for these studies. So MIMIC is very dominant in this context. And this now leads then to the key issues and challenges that we discovered in our systematic review. So one big issue in comparing these more than 20 studies to each other is heterogeneous label definitions. So which definition was used to define sepsis? And some of these definitions are even insufficient for the early detection task. So sometimes there are even like data sets where someone used billing codes from the hospital to label like a patient as someone who has had sepsis during the stay. But this very coarse information, whether the patient has had sepsis or not during the entire stay, is not enough for an early prediction problem. In an early prediction problem, you need information on the actual onset, or the actual time point at which sepsis started. So there we have this huge heterogeneity, sepsis 2, sepsis 3, labels for the entire time series, and so on. So sepsis is not defined in the same way in these different studies. There is heterogeneity there. The other one, and that's actually more scary, I would even say, the other big problem is that even if one uses the same definition, sepsis, let's say sepsis 3, the concrete practical implementation of that definition varies between studies. So we have seen papers that claimed to use the same data set, MIMIC, that claimed to use the same definition of sepsis, sepsis 3, and still ended up with different numbers of sepsis cases. And they differed not only by a few hundred or so in a data set of thousands, but they differed by one order of magnitude. So in one case, we had more than 15,000 sepsis cases in one paper, and in another data set only a few thousand sepsis cases with the same data set and the same definition. So that gives you an idea of how complicated even the practical implementation of these sepsis definitions is. Now, to solve this problem, of course, or to at least improve upon the current state of the art, of course, it would be great to have the code that was used for these annotations of time series with sepsis labels. But this is another challenge and problem in the field. So the code for the label extraction, let alone the code for the predictions, is often not available. It's often not published with these papers. So there's not a lot of software that accompanies these 20 papers. Then there's an issue of circularity that one always has to carefully consider. When you use the vital signs of a patient and the data of a patient to define when sepsis started within the time series, then you must be careful not to use the same observations for predicting sepsis, because that would be circular. That would be a self-fulfilling prophecy if you use the same variables to say, I define sepsis onset after 10 hours, and then you use the same observations that were used to define the sepsis onset after 10 hours to predict sepsis at 10 hours into the stay in the intensive care unit. That's another circularity one has to think about. And then, of course, another big question is, what are controls in this context? So if you have sepsis patients with an onset time, with a point of time during the stay at which sepsis started, what is the control then? Like a person that did not develop sepsis, but what is the onset the onset time point, or the pseudo onset time point for a control then in this context? That's a very difficult question to answer. In fact, we'll see in a minute, different strategies have been used for that. And this becomes apparent when one looks at another element that differs between these studies, namely the evaluation strategies that have been chosen by these different papers. So out of the 21 papers that proposed a new approach, 19 did what we would call an offline training plus horizon evaluation, and 2 did an online training with online evaluation. And I'll elaborate on these 2 strategies now in the following. The first one, the offline training plus horizon evaluation. What happens here? So what happens is that we do what we described before. So for a case, this is a person that develops sepsis in the ICU, we define a time point of sepsis onset. So that would be, to make it clearer, that would be the time point in a time series at which all criteria of the sepsis 3 definition are fulfilled. This is the point in time. This is what we call sepsis onset. And then we define a feature window, some y, I used the x before, so let's say y hours before that sepsis onset. And that feature window represents our patient, our case in that case, in this scenario. Now, the control patient, in the control patient, as I just explained, there is no onset. The control patient doesn't develop sepsis. So we need what I would call a pseudo onset of sepsis, such that we have also a reference point in the sepsis, in the non-sepsis patients, in the control patients. So one strategy is shown here on the right. You choose a random point. This would be the pseudo onset, or the matched onset, we call it here, for that is randomly chosen along the time series. One other strategy is to place the pseudo onset at the absolutely same time as in one corresponding case. So if in that particular case, sepsis happened after 10 hours in the ICU, you would put the pseudo onset in the absolute strategy also after 10 hours in the ICU for this matched control patient. The matched pseudo onset, according to the relative strategy, would say if the sepsis onset in this sepsis case here happened after 60% of the time that that patient stayed in the ICU, you would also put it at 60% of the time that this control patient stayed in the ICU. So these are three strategies that you can observe in the literature. It's not always exactly defined in the papers which of the three strategies was used. But this is this kind of pseudo onset definition that is commonly being used. And then when you have that pseudo onset for the controls, you can also define like a feature window there, like a sub window of these time series that is also Y hours before this pseudo onset. And then you have time windows that represent cases, and you have time windows that represent controls. Now in the horizon evaluation, what you do is you vary Y. So you look at these feature windows that are Y hours before the onset, and you vary Y from 10, 9, 8, 7, 6, 5, and so on, hours before onset up until the point of sepsis onset. And then you can examine, for example, how many hours in advance for which value of Y can you predict sepsis accurately. So that would be a typical setup here. That would be the strategy one that is completely offline. It's absolutely retrospective, as you can see here. So you look at the time series retrospectively. You compute when was the onset of sepsis, and then you choose a feature window prior to that, which is the input to your machine learning algorithm. Strategy two is much more online. So now here the classification is done by labeling every time point in the time series. So every time point gets a label. We not only define a sepsis onset, but we also define a class label, like a phenotype, for every time point in the time series. And 0 here means being in a normal state. 1 means being in a septic state. And we put a window around the sepsis onset that we have computed before, and within this window, you are in a septic state. So all time points that fall into this window are septic time points. That's when the patient is in an acute septic state. And so in this time window around the sepsis onset, all points are labeled as 1, as being septic. Outside that window in the cases, and for all time points in the controls, the class label is 0. So now you can make a time point based classification. For a test patient, you can now hide this label or assume that you don't know this label and try to predict whether, or to classify whether that patient is currently in a septic state, 1, or in a healthy control state, let's put it, say, 0. This is the online scenario. Of course, both have their difficulties and their advantages. There is a notion of earliness, as I've explained in this horizon evaluation that's being done in the offline training. Very problematic is how exactly you define these pseudo onsets in the controls. The online training is much closer to the way how you would really use such a system in the clinic. In the clinic, you would put an alarm box next to a patient, and that alarm bell should go off when the patient becomes septic. And the second strategy is much closer to that. Of course, one difficulty here also exists, that is the size of this window around the sepsis onset. So how big do you make this time window within you consider the patient to be in a septic state? And there's yet another big problem in the state of the art in sepsis prediction, and that is the lack of external validation. When you look at these 21 papers I mentioned before, then more than 85% of them do not use any external validation data set. So they only present results on one single data set, and that's in most cases mimic. Why is this the case? It's not laziness in any way. It's the difficulty to even get all the variables that are necessary to define sepsis-free, for example, for one data set. So even getting this for one data set can be a major effort. So then most researchers avoid to go through the same effort again for a second data set, and often one may even have problems in obtaining all the necessary variables to define sepsis in a second data set. That's why most of the studies are one data set based, but that goes then hand in hand with us not really knowing about the generalization abilities of these sepsis detection algorithms. That is very unclear whether something that works on mimic then also works on another data set. One data set, for example, would be HIRED, a Swiss ICU data set. We also contributed to its creation. So when you have good results on mimic, it's hard to tell whether you would also do well on HIRED, for example. And that motivated a large study of ours to harmonize the major public ICU data sets. And that was joint work with the labs of Nikolai Mainzhaus and Peter Bühlmann when I was still at ETH Zurich. And this work is currently under review and will probably, hopefully, appear soon. It's predicting sepsis in multi-site, multinational intensive care cohorts using deep learning. We harmonize the major public ICU datasets in this work. So let me give you an idea of what datasets I'm talking about here. So there are two big ones from the US, the aforementioned MIMIC and EICU. And there are two big ones from Europe, AUMC from Amsterdam, and the HIRED database from Switzerland that I also mentioned before. Emory is another dataset from the US, but smaller, with a smaller variable set that was created for a competition. So for these five datasets, we performed data harmonization. We extracted the same variables and did variable mapping between the different hospitals, removed artifacts, harmonized the units to then create a harmonized data set of 156,000 ICU stays. And if you concatenate all of these stays, it's more than 783 patient years that we are talking about. And these includes 26,000 sepsis stays in this harmonized data set. Sometimes we speak of 5 data sets. When we look at these 5 here, sometimes we only speak of 4 because in Emory, not all the variables were available that we have for the other 4 data sets. So often we only consider the big data set where we have full availability of all features that would then exclude Emory. Now, we then, after harmonizing these data sets, we then aimed to do the biggest external validation of sepsis prediction algorithms that has been done in literature so far. And we did so using an online strategy, what I described before, that we computed the sepsis onset shown here according to the sepsis-free criteria. We defined a time window around this onset, which goes 6 hours into the past and 24 hours into the future. This is the time period in which we deem someone to be in a septic state. That was based on discussions with our medical experts and collaborators. And outside that time window, the state of a patient is 0. And then we could do what hasn't been done in literature so far. We could train on each of these big 4 data sets now. Now, this is here focusing on the 4 big ones. We trained on Amsterdam, HIRED, the 2 American data sets, shown here in the rows. And then we predicted on those shown in the columns. And this is now the area under the ROC curve, so our ability to distinguish septic from non-septic, using the best algorithm that we developed, which was a deep learning based method on that used self-attention. And what can we see here? Well, we see here that internal validation works well to very well. So when we look at the diagonal here, training on Amsterdam, predicting on Amsterdam, 92% area under the ROC curve, 83 for HIRED, 83 for MIMIC, 80 for EICU. This is what we would call internal validation. You train and you test on subsets of the same data set. Now, when we do really external validation, then we have to look at the off-diagonal entries here. We still get moderate to good results, I would say. So the areas on the ROC curve we reach here are certainly all non-random. The worst performance is predicting on MIMIC after training on HIRED at 65%. The best one that I can see here is training on HIRED, predicting on Amsterdam is 81% area under the ROC curve. So still a good result that we achieve here in external validation. There have been attempts to validate sepsis prediction algorithms across clinics in the US that gave much worse external validation results than what we show here. So these are actually quite good and quite successful. Statistical, of course, statistical and retrospective external validation results for sepsis. What worked even better in external validation was what we called the pooled N-1 approach. So what do we pool here? And what is N-1? Well, the idea is that if we want to predict on Amsterdam, for example, then we build one predictor, one classifier each for each of the other 3 data sets. So N is here 4 for the 4 data sets. So for the N-1 is 3. For the other 3 data sets, we build one predictor of sepsis each, one predictor for HIRED, one for MIMIC, one for EICU. This classifier we then use on every time point of a new test patient that we look at. And we always take the most pessimistic prediction. So we believe the classifier that currently gives the highest risk for that patient to develop sepsis. And if you do that, if you pool 3 separately trained classifiers and always take the most pessimistic one as the winner among these 3, then we have external validation results that lie between 72% area under the ROC curve and 84% under the area under the ROC curve. And these are really good results for external validation. A nice thing and elegant aspect of this is that we do not even have to share the data between the different hospitals. We train one classifier on the Swiss data, one on MIMIC, one on EICU. All we share is the predictions in the end. So the patient data in itself doesn't have to be shared in this pooled N-1 setup. Now I'll summarize the remaining part a bit faster to also have some time left for the antimicrobial resistance project. So we tried various classic statistical, classic machine learning approaches, classical intensive care scores for predicting sepsis here, but also modern deep learning architectures like a self-attention based approach. What I want to highlight is that in such an early prediction task, of course, it's not only about precision and about recall, how your how you should judge the quality of your method. It's also earliness. So it's 3 dimensions, it's a bit different from the typical classification problem. Can you make a precise prediction? Can you make predictions with high recall? But can you also make them early? So you have to evaluate your classifiers here on 3 different dimensions, you could say. And we see, for example, here in this example, that one of the best performing methods here, this is attention here, the dark green one, that gives high quality predictions 4 hours before sepsis onset in the internal validation. But when we move to the external validation to a different hospital, then it's only 1 hour before sepsis onset that we get high quality predictions. So we not necessarily lose in precision, but maybe also in earliness we may lose when moving from internal to external validation. We did an analysis in terms of Shapley values, which parameters contribute to our predictions, so blood pressure, heart rate, urine output, and other variables that in general make sense as being related to sepsis here contribute to our prediction. Now, this data is about to become public when the paper goes online, and it's one of the first, if not the first example, where one could statistically, on a large scale, validate and even successfully validate a sepsis prediction in a retrospective in silico study. Of course, the limitation is also this fact that it is in retrospective in silico study. Certainly, the future has to then bring the prospective in clinic studies, but this, in our eyes, here is also a big step for this field of machine learning for sepsis prediction. We did similar work that appeared in 2020 in Nature Medicine for circulatory failure. This was a collaboration with the labs of Tobias Merz from Inselspital Bern and Gunnar Rätsch from computer science at ETH Zurich. And this followed a similar principle, just a phenotype. The condition here was circulatory failure. Now, and this is something that I've been doing up until early this year. Pablo mentioned this in his introduction. There's a big European effort in Switzerland to do sepsis prediction in Swiss university hospitals. So under my coordination and that of Adrian Egli from now University of Zurich, we harmonized Swiss ICU patient records from the Swiss university hospitals across all Swiss university hospitals, and we have collected over 18,000 health records from intensive care units across Switzerland as part of this program, and built the infrastructure for sharing such data across Switzerland. In the second phase that I'm not coordinating anymore because I moved to Munich to become expert director, omics profiles of these patients are also being collected. So in this phase 1, we primarily collected vital signs, lab tests, health history information, not detailed omics information about the patient. So what is nice about sepsis prediction is that it includes many aspects that are so relevant in machine learning in medicine. So this automated diagnosis, this finding the onset of sepsis is a major point. The biomarker discovery, so which variables are predictive of sepsis. This whole biomedical data management, I only quickly touched on that when I mentioned the Swiss program. Of course, it was a huge effort to build this infrastructure for sharing this biomedical data. You can extend this in various directions. You can look at molecular data. You can look at clinical validations of the predictions. You can look at data before people enter the ICU. You can look at the long-term effects of sepsis. So there are many ways in which you can extend this work, and many of the approaches and insights that we have gained here apply to other clinical prediction tasks as well. So sepsis prediction remains a highly exciting topic in machine learning in medicine. And now the remaining few minutes I will use to summarize the second part. I won't discuss it in the same depth. And the second part deals with a related problem from machine learning for microbiology, for clinical microbiology, you could say, namely antimicrobial resistance prediction. So antimicrobial resistance is, like sepsis, a global health threat because antimicrobial resistant pathogens, those that do not respond to antimicrobial treatment, often antibiotic treatment, increase morbidity, mortality, and of course, healthcare costs. And as we said for sepsis before, rapid treatment with effective antimicrobials is key to improved outcomes and optimal stewardship. And even the World Health Organization has recognized that. And there's a strategic priority to find ways to fight antimicrobial resistance. So we wondered, can machine learning, can AI play a role in that? And now I want to describe to you, and that's actually the key thing I want to do now in the remaining time. We defined with our collaborators around Adrian Ekli from University Hospital Zurich, we defined the exact point in the clinical workflow at which we could support the clinician with AI. In order to understand this optimal time point, I have to explain the classic workflow to you. So when you come to the hospital, at least in Basel, with an infection, but this may be similar across the world, in fact, then a sample is collected, like a body sample, body fluid sample, or blood sample is collected. Then the pathogen is grown on an agar plate for 12 to 24 hours, and then it undergoes a multitoph measurement. So you measure the presence of protein fragments in that sample. And you do this in order to identify the species that is causing the infection. So the species of the pathogen is identified with this multitoph technology. That's a mass spectrometry technique that is being used there. Then you let the pathogen grow for another 24 to 72 hours and determine resistances. So you expose the pathogen to different antimicrobials and different antibiotics and see to which of these it is resistant. And then you give that information back to the treating clinician, who then chooses a specified antimicrobial treatment. So as you can see here from this workflow, there is a delay in this second part, clearly, where you experimentally determine the resistances of these pathogens. And we wondered whether we could speed up that second part, whether we could take the multitoph measurement, this mass spectromic, this proteomic measurement, and predict the resistance directly from these mass spectra, rather than going through this experimental determination of the resistance phenotypes, thereby saving 24 to 72 hours, because making a prediction on like a multitoph mass spectrum can be done in seconds. And this is now what we built here in this big collaboration. I won't go through all details here now, but let me highlight a few things. So we gathered data in this collaboration with Adrian Ekli from hospitals and laboratories in northwestern Switzerland. We compiled a data set that we in the end called the Database of Resistance Information on Antimicrobials and Multitoph Mass Spectra, short DREAMS. And as you can see here from the numbers below, DREAMS has 4 subsets. These are these different hospitals and laboratories in Switzerland. In total, we have over 300,000 mass spectra in this data set. We have over 700,000 antimicrobial resistance phenotypes associated with these mass spectra. So that's the largest clinical data set that is publicly available for this purpose. You can go to our paper and follow the link and download this data set in seconds if you're interested. So, and you're very welcome to do so. This slide here summarizes the steps that we had to go through. Of course, there was a huge data collection and filtering effort here, a huge effort of processing the spectra, and then of antimicrobial resistance prediction using machine learning. And what we found out is that for very relevant and highly prioritized species antibiotic pairs, we could predict with rather simple machine learning methods antibiotic and antimicrobial resistance phenotypes well. So we reached 74 to 80% area under the ROC curve with the best methods on these combinations of species and antibiotics shown here on this slide. We then tried to find out why this worked well, and it's to some degree surprising because the mass spectra are often, or in most cases, not fine-grained enough to really pick up the resistance mechanism that causes resistance to an antimicrobial. But what we found in some experiments is that the classifier picks up information about the pathogens being related to each other. So sequence, excuse me, not sequence, but lineage similarities between the pathogens. So it's a measure of relatedness. So the algorithm uses the mass spectra as a measure of relatedness between samples, thereby implying that related lines will have similar resistance phenotypes. So we found that positions that indicate relatedness between these clonal lineages were important for the classifier in E. coli, for example. And the validation between different hospitals worked, I would say, moderately well in some cases. In others, for other species, antibiotic combinations, it failed. It was really bad. So validation across hospitals is a big issue here. So you may ask, but if it doesn't generalize, how can it be helpful? Well, in this particular setting here, a nice and interesting aspect is that there are so many mass spectra being measured in these hospitals and laboratories every day that you can locally create a data set. It's not impossible. In many other machine learning applications, it may be to get at every site like a new training data set, but here there's a lot of clinical routine data being collected for identifying pathogen species. And you could, in principle, use that clinical routine data to build new predictors, to have new training data sets all the time, even if the predictions do not generalize across different sites. So that's special about this setting here. Of course, it would be much easier, even logistically, regarding infrastructure also, if you could build predictors that generalize across sites. But the possibility of retraining predictors per site still exists. We looked at what the best strategy is to combine classifiers depending on which sites they have been trained on. So best strategy turned out to be that if you want to predict on Tremes B, for example, which is not the biggest data set, is that if you combine then training data from Tremes B with the large data set from Tremes A. And then you got some of the best results. Also, there's a temporal validation issue. These mass spectrometry machines, the multitoph machines, they get recalibrated, or the lab technician changes over time, and this may also have slight impact on the results. And you can see here on this plot that the performance of the predictions gets worse the larger the time difference between the time point at which the validation, the test set was collected, and the time point at which the training set was collected. So the training set was here collected at the right end of this timeline here, and the further you go left, the further you go away from that time point, the worse the area under the ROC curve gets for the three pathogens shown here. And then, and this I'll only summarize quickly, we also had an infectious disease specialist analyze whether the predictions we make here would have made any difference on patients that had been treated in the hospital. And we found 63 patients in which this could be examined. And in 54 of them, our predictions would have made no change to the suggested antimicrobial treatment. In 9, it would have. Out of these 9, in 7, we would have de-escalated the antibiotic therapy. In one, we would have continued the antibiotic therapy. And in one, based on the algorithm's advice, we would have escalated the antibiotic therapy. Only the latter one would have been a false decision. So in 8 out of these 9 cases where the algorithm would have made a difference, in these 8 cases, it would have been to the benefit of the patient. And that's a nice outcome here. So the algorithm can be helpful in the treatment. I also want to mention here, when carefully analyzing these results, there are also cases where the doctor compensates for a mistake made by the algorithm. So the very interesting finding of this is also that the combination of doctor and algorithm is better than each of them alone. That's also a very nice result that came out of that. So to summarize and to conclude this second part, this was the first large-scale study to analyze the clinical potential of multi-TOF mass spectra being used for antimicrobial resistance prediction. The feature interpretation, as I showed, was congruent with the literature. Transferability, external validation remains a challenge. The dataset is publicly available, so is the code we use to analyze this data. So with this, I want to thank you for having me today. I want to thank my lab members who contributed to this work. So Caroline Weiss was the first author on the second project, and there's Michael and Bastian were the first authors on the systematic review that I showed in the beginning. This here is still ETH because this was the work that happened over the last 5 to 6 years at ETH and is now to be continued at the MPI of Biochemistry in Munich. Thank you, and I'm looking forward to your questions.
Thank you very much for this very nice talk. So yeah, time for questions. So feel free to write your name in the chat box and I'll give you the turn to speak. Any question? Any questions from the audience?
If there are none yet, but maybe later, you can also send me an email. My email address is easy to find. I think it's even in the announcement of the talk.
Well, in any case, I have few questions. Well, I have many questions, but we don't have time for, for all of them. Well, the first one is related with how is that related to the first project or strategy you mentioned? How is that more than 85% of the studies do not use any external validation data set? What could be the reason for that? Because from the machine learning point of view is something.
Yeah, I'll go back there. It's so hard to get one data set on which you have all the variables for doing the predictions and all the variables for doing a proper annotation of sepsis onset. That when you have one, I mean, you have already spent so much time and effort on that you don't, so then very often this is then not repeated for a second data set. So it's really a lot of effort. And that's why we said, oh, we can really greatly contribute to the field if we create this harmonized data set so that you can so that you don't have to do this this harmonization effort, that you can just use our data set, our harmonized data set, and at least get a statistical retrospective external validation of your predictions on 3 or 4 other data sets when it works on one. And it is a very difficult task. So I mentioned also that it's very hard to implement the sepsis 3 definition, and even there are problems on one single data set. And then the question is, how do you do this on a second data set? So these are all big obstacles. And I would say the data curation effort is just so big on one data set that it's often avoided to do it on a second data set. But this dilemma we wanted to solve, the dilemma that due to that we cannot say anything about generalization. This we wanted to solve by harmonizing these data sets and by making this available to the research community.
Okay.
Yeah.
Perfect. And well, another question I have is that, at least in my experience, medical problems often require access to sensitive personal data, personal information. So in this sense, have you developed privacy-preserving machine learning algorithms that keep the patient's details secure without affecting performance? Or this is not particularly important in this application? Or what
It's
could you
extremely
say?
important in both, but there are different solutions in both. So and there's a third one that we do not use so far, but that others use. So the third one is, I think what you alluded to, that you have like distributed machine learning. You build privacy-preserving distributed machine learning approaches where you predict like the same condition across hospitals, but you do not exchange data between the hospitals. That's one line of research. There are excellent colleagues who work on that, who build like distributed machine learning algorithms that do not really share hospitals between data between hospitals, excuse me. And but what we did was the other strategy. In the Sepsis project, I'm going there, we harmonized data all over Switzerland. And we really have like one central server where all the harmonized data comes together. So here only the infrastructure is patient privacy preserving and the way how we transfer the data from one hospital to the central node. But this is centralized, this setup. Here it's also all secure, but we do not develop special algorithms that are privacy preserving here. Of course, if we publish the data, we anonymize them to a degree that it is allowed to then publish them. We would never publish like the raw patient data in any way. But here it's all centralized in this approach, in this personalized with sepsis study. Now in the antimicrobial resistance project, there so these mass spectra, they come from the pathogen. So in that sense, they are not patient data. So then they are we could publish them without problems. It's just bacteria in most cases here. The only thing that included patient data, but this we never had access to, was this retrospective clinical case study that the doctor performed. But this happened within the hospital with the necessary approvals. And but in the rest of the project, we did not use patient information. Maybe you could actually improve the prediction a bit more if you had the age and gender of the patient from which this pathogen was isolated. So maybe if you had some more covariates on the patient that the sample came from, this would improve things, but of course it would make the study much harder and more complicated in terms of patient privacy. So these are the three ways here. So in one way, in one project, we did not really work with patient data in the legal sense. In the other, we centralized the data in a secure environment, and others in the field do these federated machine learning algorithms. I mean, the disadvantage of those is you have to make very strong assumptions on the format that the data has at different sites. And in the centralization effort, we have seen how much discrepancies there can be even between the same data from different hospitals. So the distributed approach is very elegant, very beautiful, very useful. Still, it makes strong assumptions on the format of the data that you combine over different sites.
Okay, well, thank you very much for your answer. Now, yeah, Rocío, go ahead, please.
Well, first, I want to thank for this great talk. And following Pablo's question, I wanted to ask, since you work in different countries, if you have an idea of the different, how do you say this, politics they use in different hospitals, public hospitals, private hospitals, about the use of the data. How do you convince a hospital to give you the information? What are the tips and tricks to do that?
There are no tricks, I believe. What is extremely important, besides, of course, like following the law, is a relationship of trust between the data scientist and the clinician. And as you can see, as you have seen here, these two talks were largely collaborations between me and the lab of Professor Ekli, who is now in Zurich. So we have a long-standing tradition of collaborations. We trust each other. And it's often the basis then for exchanging such data. Even we exchange data that is not privacy protected, like this pathogen bacterial information. But still, I think that's very important, and also like a social skill in this very mathematical or computational branch of research. A social skill is to build a good relationship of trust to clinical researchers. There's always, I mean, a legitimate worry, I would say, among clinical researchers that they spend a lot of time on collecting high quality data, on curating high quality data, and that then some data scientists may come along and take the data, analyze it, publish it without acknowledging them. This is a constant worry that is also legitimate. You have to build a relationship of trust such that the other side knows that this will not happen, that both sides do fair play. I mean, this probably holds for all kinds of scientific collaborations, but in this clinical scientist and data scientist collaborations, it's extremely important also. Yeah.
Okay, and what about the fear of the medical doctors and nurses and people working in hospitals about the use of AI that lately have been complicated? Do they try to get information about the explicability of the work you are doing, or they just want results?
I mean, they want to have interpretable machine learning. They want to know why a certain decision is taken. To answer the second part of your question, in the first one, you mentioned this threat from AI. I mean, there one has to define what we mean by threat. I mean, one threat would be if AI starts taking life and death decisions, and I mean, that would be terrible. There must always be a human level in this decision, and that like a human doctor checks what the machine is doing. I showed this example where the combination of doctor and machine is better than any one of them alone. I think that's the model for the future. And of course, we must regulate these machines such that they don't take really life and death decisions without involving a human expert.
Okay. Oh, thank you so much for your talk.
You're welcome. Thanks for your question.
Thank you very much. Well, I have a very quick question. If there are no other questions from the audience, I mean, I don't want to monopolize this conversation, so please feel free to ask. Feel free to write your name in the chat box in case there's any question. No? Okay. So, I mean, related with the last question asked by Rocio, model interpretation, if I'm not wrong, you did a quick mention in slides 27 and 50, but if possible, I'd like to know more about model interpretation regarding project 1, the first project you presented. What kind of explanation did you provide? In slide 27, I guess what's the explanation?
Is this the Shapley values, like which vital signs or which lab values and so on contribute to the prediction? To the prediction. That's what's shown here. So, and they are ranked by that contribution. So the topmost is the most important one, but many contribute. Yeah. And I mean, many of these may be linked to the weak, predictors of sepsis that I mentioned in the introduction. I mean, the art of deep learning is then often to combine them in a nonlinear fashion to get a stronger prediction of sepsis onset. And I believe that's what's happening here, and when we are combining different values into a stronger prediction of sepsis. Okay. Yeah. I mean, that's one way. Of course, the more complicated the models get, the deeper they get, the harder the interpretation. That's absolutely the case. Yeah. And so just my final question is, are these two
models, these two predictors, are being used in the daily clinical practice nowadays, or not yet? Or
not yet, not yet. So in this, the second one is much closer to being used in the clinic, because so just is a big word here, but we just have to implement this in a clinical laboratory, that a prediction is being made based on a mass spectrum. So the mass spectrum is measured anyway for identifying the species. So what we then need is, and we are looking for people for doing that at the moment, someone who implements then that a prediction is being done about the resistance phenotypes for that pathogen. So this is one thing. The other one, and there is doing a prospective validation of sepsis onset. There were plans to do so when I was in Switzerland. Now, due to my move, these are a bit halted at the moment, but it's certainly on my agenda to take this to the clinic, to the ICU, to have really an alarm at the bedside, and to see whether having such an alarm, such a prediction of sepsis, then changes outcomes like mortality in the ICU, length of stay in the ICU, and so on. This is a multi-year effort, the latter one. So the first one maybe can be a 1 to 2 year effort. The other one is a 3 to 4 or 5 year effort, I believe.
Okay. Well, so thank you very, very much, and well, congratulations for all your work and all your team.
Thank you very much. Thanks for the nice question and answer session.
Okay, so we can close here the session. Thank you very much, Karsten Borgwardt, for your very interesting talk, and thank you to all the attendees for being here and for the interesting questions.
You're welcome. Thank you. Bye-bye and have a good evening.
Bye-bye.
Bye-bye.